improved methods for cDNA cloning. Although different methods are available for the cloning of cDNA that were proposed by different workers at different times, and here we will focus only a few outlines. So the hairpin method that we discussed in the last topic, it has certain disadvantage. The reason is that when hairpin loop structure it is formed, and finally when it is degraded or removed by S1 nuclease, then some portion of the DNA at 5' end it is also degraded. So because of this reason, this method has been superseded by other techniques, and where we can direct the synthesis of second strand in a separate reaction. It will be cleared by this diagram. So in this diagram, that showed the improved method for cDNA cloning, and it was developed by Land and co-workers in 1981, whereas the Maniatis method was in mid 1970s, and it is an improvement over that traditional hairpin loop method. So here messenger RNA, usually it contains adenine residue stretch at 3' end. Then it can be subjected to enzyme reverse transcriptase and 4-DNTPs, so that first DNA strand it is being synthesized here. So this is the messenger RNA that act as a template for the synthesis of the first strand of the cDNA. Then in the next step, this cDNA it is subjected to terminal transferase in the presence of cytosine residues. Now it will add cytosine residues at 3' end of the cDNA. Now these two strands, they are treated with alkaline sucrose gradient, and it will result in the hydrolysis of the RNA, and it will also recover the full length cDNA. So alkali will remove the messenger RNA. Now we have a separate strand of cDNA. Now it will be used in a separate reaction to direct the synthesis of the second strand. So onward, when first strand it is being synthesized, then it is provided with guanine residues, and it will result in the second strand biosynthesis. So after the synthesis of duplex cDNA, now it is ready to insert into a suitable vector by homopolymeric tailing. Sometimes it is advantageous if the insertion of the cDNA it is correctly, so orientation if it is correct. So for this purpose, another type of strategies have been developed that are based on hairpin method. So traditional hairpin method have also been used to insert the cDNA in correct orientation. So for this purpose, synthetic linkers have also been used with cDNA. For example, this diagram, it is another method for directional cDNA cloning. So like the usual hairpin method, again the messenger RNA, it contains polyadenylated residues at 3' end, and thymine primer it is used, as we discussed number of times, to direct the synthesis of the first strand of cDNA. After that, DNA polymerase Plano fragment it is used, and if you remember, this DNA polymerase it lacks 5' to 3' exonuclease activity. So again, this hairpin like structure it is used to direct the synthesis of second DNA strand. Then second strand biosynthesis it is followed, and here it is an improvement that linker molecules they are ligated, and these linker molecules it contain restriction site for Sol1 enzyme. So linker molecules are added at right side, and that contain the Sol1 restriction site. And then the left side are 5' and they are digested with S1 nuclease, and then again provided with DNA polymerase colony fragment in the presence of all 4 DNTPs, and we have a duplex cDNA that contain linkers with restriction site for Sol1 enzyme. After that, this duplex cDNA, it is again provided with or ligated with linkers that contain EcoR1 site. So on the left side are 5' and linker molecules they are added, either on the right side or the left side. And then they are cut with Sol1 and EcoR1 so that we can produce sticky end at left and right side. So at right side we have sticky end for Sol1, and on left side we have site for EcoR1. And then it is inserted into a vector that is being fragmented with Sol1 and EcoR1. So ligation it is there, and after ligation it is used to transform suitable host organism. This is another method for directional cDNA cloning, and here we can have some variations, like usually again messenger RNA with 3' polyadenylated, and it is used, and here the primer that is based on a stretch of thymine, it contains a stretch of another nucleotide bases that contain restriction site for Sol1. So in addition to thymine residue, it also contains sites that contain Sol1 restriction enzyme sites, and then it is subjected to reverse transcriptase in the presence of 4 DNTPs. So first strand biosynthesis it is there. Now in addition to thymine primer, now some stretches of DNA or nucleotide bases are also there that contain restriction site for Sol1 enzyme. Then first strand it is subjected to DNA polymerase colony fragment that lack 5' to 3' exonuclease activity in the presence of 4 DNTPs to direct the synthesis of second strand. Now second strand it is being synthesized. Now on the right side it contains linkers molecule that contain Sol1 restriction target site, and then it is provided with EcoR1 linkers here. So on the left side linker molecules for EcoR1 they are added, and then they are again cut with Sol1 or EcoR1 restriction enzymes, and now they are ready to insert into a suitable vector that is ready for transformation. So these are a few improved strategies for cloning cDNA into a suitable cloning vector. So number of other strategies are also there, but we have just focused on a few strategies here.